Thank you so much for joining me. Today, we're talking about the role of Bifidobacteria longum and its altruistic role and keystone, being a keystone flora of the human gut and the guts of many mammals on Earth. I'm a biohacker. I don't know how many biohackers are in the house. Maybe raise your hand if you have an Oura ring or track your heart rate variability or track your 16S gut stool. Okay, you're all like me here, a poop nerd. So I formulated a probiotic. We have a couple SKUs. One is Bifidomaximus, and it has one of the highest potencies for Bifidobacteria longum on the market with 40 billion per dose, or it comes in a powder as well, which is 100 billion per scoop or 40 billion per quarter teaspoon for Bifidobacteria longum. I'm focusing on Bifidobacteria longum for today's talk because we don't always look back at evolutionary history, and I wanted to share about how it has impacted all creatures on Earth, at least the last 100 million years. And today we'll talk about the altruistic role. It's actually a vital keystone flora, and some may even consider it an apex predator. We're going to review its evolutionary history over the last 100 million years for both the birds and the bees, as well as humans and other hominids and primates. The phylogenetic core microbiota will be discussed, including that of the ancestral bee, as well as ancestral and modern humans right now, and long-lived humans. How many of you might want to have longevity and long telomeres? Great, I'm with you. And how does Bifidobacteria longum benefit us for longevity? There's some great studies, but most of it's kind of speculative, but it'll be really interesting to look at that. And how I'm a functional medicine practitioner and regenerative medicine adjunct professor, faculty, and I love biohacking. So I'll go over some cases where we're doing advanced stacks. I call this stacking and hacking. And some include some peptides, if some of you are familiar with peptides. So why look at Bifido over evolutionary time? Well, human Bifidobacteria longum may have descended from the bee, Bifidobacteria asteroides, and this appeared about 70, 80, 90 million years ago. Bifido has actually been around through some deep studies, probably even like up to 100 million years ago. And it jumped hosts. Every time it jumped hosts, it apparently, according to studies and some theories, the species developed some new gifts, some new traits, and it persisted in the gut of the new host. They're not sure who was the original host, but it could have been a swine, a pig, and then it jumped to insects like bees, and then it stayed there. And it stayed in the bee gut, apis, and the social tribes of bees for at least 80 million years. And what's so fascinating is that through comparative genomic analysis, there suggests that a long history of coevolution between Bifidobacteria and their hosts, whether it's animal, bird, reptile, or insect. It kind of makes me curious if Bifido was also found in dinosaurs, because that's the time frame, right, before that extinction. And we're kind of going through another extinction now. I call this the sixth extinction, but some of our original bifidos that are found in healthy Europeans and healthy moms, they're no longer seen in the Western gut in babies or breast milk. So it's a little scary. But when there's co-speciation that happens according to evolutionary history and studies now, when there's a new species that comes out, there's actually a new strain of bifido that goes with it, and it persists according to genomic studies. So there's coevolution with social animals, creatures only. Like there's 3 tribes of bees, honeybee, bumblebee, and stingless bees, some birds, and then mammals. And what's so special about Bifidobacteria longum subspecies longum is that it is in a lot of animals and mammals that take care of babies and have caregiving roles, just like social bees. And what's so amazing about another Bifidobacteria longum subspecies, the infantis, is that it appears that it co-evolved very specifically for the human mother-infant relationship, the dyad. In fact, several studies show through genetic analysis, the species and strains are exactly the same. And this is to be expected, right? So Bifidobacteria, it's one of the most ancient bacteria coming from the phylum Actinobacteria. They're non-sporulating, they're non-motile, they are gram-positive. The bee ones are microaerotolerant, but most all other species, there's probably like 100, I can't even keep up. They appear very anaerobic. And how are they transferred? Well, in bees, let's go back to the birds and the bees, okay, if you guys don't mind. And we're not talking about sex, unless you want to, I'm kidding. So when we go back to bees, when they're first born, like from the larvae, there's eclosure. And there's no gut microbiota in that larval, and it gets colonized. There's engraftment. And researchers aren't totally sure, but they think it's not just from oral trophallaxis alone, where the nurse bee is feeding the larva either honey or royal jelly or whatever the food might be. They think there's also transfer through the contact in the tribe and possibly oral fecal and other contact. Bee tribes are very intimate. And this is a lot like humans too. Through birth, there's defecation, sometimes often, right, of the mom on the baby. And one of the first colonizers in a baby, human baby, and probably farm animals and other animals, mammals, is E. coli. It's first or second next to the bifidos, Bifidolongum infantis and the others. So we're not that far or different from the bees. Babies are born, unless it's C-section, they're born sterile in the gut microbiota, unless there's mom had health issues and a lot of permeability, eclampsia or some complications, but generally it's a lot like bees. Now, what happened 80 million years ago when the social bees deviated and had co-speciation with their bifidobacteria? Well, they acquired some really amazing things. They separated from wasps and other bees, and they became social. A queen developed, and so interesting, the queen bee lives a lot longer than the nurses of the drones. In fact, studies show they live 10 times longer, tenfold, 20 times longer. And in one bee, the honeybee, 50 times longer. So we'll talk about in a second why that might be. But in bees, there's a lot of caring for the young. The nurse bees will raise them and feed them. She also will feed royal jelly to the queen, as well as the babies initially. There's a caste system a little bit, and there's a lot of collaborative working together, very intimate. They're foraging, they're defending the colony together, and they're also the nurse bees are feeding one another. That's a way they send signals. In our gut microbiota, we have a lot of signaling going on. Some people call it our second gut, our second brain, and that intuitive sense. In yoga, it's also like the navel and our deep understanding of what's going on around us. So a lot of things transitioned 80 million years ago. And this kind of informs us about our bifidolongum in the human gut. And the queen bee is starting to be looked at as a longevity model because it's easy to study. And what they found was one of the key microbiome features is that it had very high bifidobacteria. Isn't that amazing? As well as lactobacilli, but it's really the bifido. And in bees, it's called Bifidobacteria asteroides. So, contributing to the longevity of the queen bee, it's the food, it's the microbiome. There's a lot of things going on. So one group of researchers, Copeland, they actually took the microbiota of queen bees that live long and put it into MFBs, microbiota-free bees. And guess what? It extended their life just as they expected. And it was due to some of the flora there. Fascinating, right? And what's royal jelly? It's got peptides in it. How many of you are into Wolverine peptides or BPC 157 or other peptides like that? Yeah. They extend our life. They make our organs brand new again. And royal jelly's full of them, as well as a lot of antimicrobial things. So it would combat pathogens that are in the GI tract or whatever bees track into the hive. Royal jelly might be, and other things in the hive, like propolis and other things created by bees in their mouths and elsewhere, might be considered the immune system of the hive. So here's what Copeland showed. Bifido was a lot higher than the worker in the queen bee, was higher than the worker bee. They also had more Bombella strain and more lactobacilli. And this deviation happened, again, about 70, 80 million years ago, and 4 or 5 clades of species are in the gut core microbiota bees. It's very true even now, according to the theoretical studies where they looked at the mutagen rate and tracked this back. Now, when we go to humans, so did bees get bigger brains to do all that they do socially and hunt and forage and defend their hive? I don't know. But for humans, we definitely grew bigger brains over evolutionary time since hominids like Paranthropus or Australopithecus, right? Or all our other Homo erectus, Homo habilis, and all those that are gone. Our brain relies on many microbiota that fuel and feed and nourish the brain. And the ones on the left are the ones where studies show that they are nootropics, and they're even antidepressant or anti-anxiety, like Lactobacillus casei or Lactobacillus plantarum. And then Bifidobacteria longum. They have known benefits, roles, and mechanisms. They're just like drugs. But I would say that the non-responder rate can be high in trials because maybe they don't use a high enough dose, or the niche is already full of pathogens and these have to be cleared out, kind of like gardening in a way. But many things are known about our brain. So if we look at human trials, we look at Andrew Mahler and a gentleman named Ockman. Ockman's from the University of Texas and Mahler's from Berkeley. Any Berkeley Cal grads here? Okay, I am one. And through their research, they looked at the evolutionary history of the gut microbiota over time. They were not studying petrified poop. No, they weren't coprolites, but they were just looking at the DNA only and the rate of mutations. And they generated some theories that there was a deviation from gorillas from hominids about 8 to 19 million years ago. And why was that? When they looked at the current microbiome of gorillas, there is a big change in the type of bacteria in the gut. And they believed it was statistically relevant. So between the gorilla, which has a mainly herbivore diet, and the Homo sapiens, which are omnivorous, the changes are all listed here. There was only 1/5 the amount of methane producers, methanobrevi factor, and a great enrichment of bifido, double the bifido, and 5 times more Bacteroides, Roseburia, Faecalibacterium, Streptococcus, all these really great things, and butyrate-making Clostridium. A lot more butyrate makers. That's so fascinating, right? Now, the change for brain size between gorilla and current Homo sapiens is threefold with that doubling in bifido. And then if we look at bonobos, panicus, there was a 4 times change in bifido. When they deviated from other the chimps, they had a drop in their bifido, only half. And then you compare it to humans, it's 4 times more, and the brain size is 4 times less. Humans and hominids had an increase 4 times. So what was so special about human bifidos? Well, we live longer with bipedal movement, our hips shrunk, and women, if they didn't have a small enough baby, they would die. There was literally a choking point, right? Bottleneck. I was thinking that word, bottleneck. And breast milk got really important. Farm animals and other animals, they only had prebiotics for the gut microbiota much less than humans. Do you know how much oligosaccharides are in human milk compared to farm animals? So we have 100 to 1,000 times more. That's a lot of prebiotic for the gut microbiota of the infant, not energy glycemic source for the baby. And the brain got as big as it could for the hip encasement, right? Before a mom would burst and a bottleneck of death, right? So the brain doubles 2 times during the first year of birth, after birth. And during that time, the mom is feeding. And so that's where human milk becomes so important. And there's basically 3 things in human milk. Lactose, so poor baby if they're lactose intolerant, but babies aren't. They're designed that way. Lipids and HMOs. Do you guys know what HMOs are? It's not Kaiser. Okay. Human milk oligosaccharides. Good job. And HMOs are also Trojan horses. They are microbial accessible carbs for the baby. Not only are they food for the baby's microbiota, not the baby, they're decoy receptors for all the junk floating around, all the E. coli in mom's poop that coated the baby. Clostridium, if babies are in the hospital, they get coated with Clostridium difficile spores from nurses, mom and dad's hands, the hospital surfaces. But studies show if they have more bifido, longum in the gut, the colonization rate goes down in an opposite way. So HMOs follow a lot of benefits. They're antimicrobial in some instances, in petri dish in vitro studies, many benefits. So bifidolongum is very special from the other bifidos. It is very good at fermenting lactose. Not so great for fermenting HMOs, actually, interestingly. But you need a little in the mix because they take over after Bifidobacteria infantis comes in as the baby weans and goes to solids. So Bifidobacteria longum might be like a keystone. We know it's a keystone flora. I'm going to go over some studies in a second, but it might be the apex predator from baby and even bees 80 million years ago to currently now. And so if we think about how apex predators work, like sea otters and kelp forests or starfish in the intertidal areas, or wolves at Yellowstone, you take too many wolves out, and then there's an excess overproliferation, right, of herbivores that will chew down all the plants until the streams start breaking up and the rivers start losing their infrastructure and the fields just fall away because the herbivores are overdoing it. So now we have lost a lot of this predator with antibiotics, and then yeast and candida take over. There's more yeast infections, BV, more asthma, more atopy, more autism, more depression, more anxiety. So what happens when we bring it back? So like a garden, we can bring it back through weeding, seeding, and feeding, right? Prebiotics, postbiotics, probiotics, and everything in between, peptides. So what are some benefits? I'm just going to be very cherry picking. I'm going to show a couple weird studies, but I really like these. So in a multi-strain probiotic, 1 billion a day for babies, within 21 days, there was a near 100% responder rate for colic. How many of you have had a baby that cried endless hours? So colic is where a baby might cry 3 to 6 hours a day. That's pretty heartbreaking, right? So it's really awesome if bifido is missing, we can replace it, and that's what this study shows. Chen and his group showed within 3 weeks with concomitant breastfeeding and mixed feeding, they were able to have a high responder rate, nearly all of them, and reduce colic, crying, and fussiness. And it's very interesting. Henri Tessier in 1899 first looked under a microscope and found bifidos. And he noticed that colicky diarrhea babies didn't have this in their stools, but healthy babies without diarrhea had it. So this goes back even 100 years ago. We don't really always need a randomized controlled trial, right, to observe something that's very obvious. And then in a petri dish study, a rodent in vivo study, they were given enterohemorrhagic E. coli, 0157H7, and if they were giving a bifidobacteria only single monostrain, they were able to prevent death, even though they receive a lethal dose. Now, a very interesting ADHD study in kids that were susceptible to ADHD, they were given a probiotic, and interesting, they were given L. rhamnosus, Lactobacillus rhamnosus probiotic at birth, and they were only given it for about 6 months, and then they were tracked for a number of years after that. Well, in the control arm, there was a 70% incidence of ADHD found, but in the probiotic group, none. And then this was held even after, beyond the 6 months after they stopped the probiotic. So I find that really fascinating, the long-term benefits. And there's something so interesting about the time in childhood where it shapes the ground zero of the immune system. We hear and see these studies all the time. Multi-strain probiotics are actually a little bit better. So in this trial with rats, which of course, not all animal studies translate to humans, sadly, but they took a multi-strain and it was high dose. I mean, these little rats got 50 billion a day and plus resveratrol. And these are diabetic-induced rats. And within 4 weeks, they normalized a lot of the insulin and gut damage, and they had a low GLP-1, and then they normalized it. How many of you love GLP-1s or for your friends or whatever? I've heard now of GLP-1 or Ozempic divorces. Either the man or woman looks super hot after Ozempic, and then a divorce happens. I think that's so funny. So what is the function of Bifido? Well, it does everything. It does everything but clean your windows and drive your car. No, I'm kidding. It does everything. So they're first colonizers, they're pathogen resistant against, again, it's the Trojan horse and decoy system, but they're also directly antifungal and antiviral and antiparasitic, even plasmodium. They're antibacterial, as we know, very anti-E. coli. But again, I think dose makes the poison, or actually in this case, the panacea, the solution. They're immunoprotective for neonates who have really no immune system except for the mom's immunoglobulins, like IgM. They help break down all kinds of foods in the breast milk, as well as mucin that coats their bellies, right? And it will feed the neonate by breaking some of these foods down to monosaccharides and disaccharides that they can consume. It's not very much for glycemic load, but they feed the symbiont gut commensal. So we'll talk about cross-feeding in just a second. And there's benefits of increasing the permeability and reducing hyperpermeability. So there's cross-feeding. They also make a lot of acetate. Acetate is vinegar. It's very antimicrobial and antifungal. And what's so amazing is on the outside shell of Bifido, they have EPS. It's exopolysaccharides, sugars for other populations in the gut to eat. So there's a lot of metabolic cross-feeding. They feed the Bifido, the butyrate makers. Their 2-carbon acetate becomes 4-carbon butyrate, and butyrate seals the gut. It's the fuel for the colonocytes. And very interesting, they also cross-feed to Bacteroides. Bacteroides is terrible at breaking down carbs. Bifido, longum, and other bifidos are huge carb eaters, and they break down larger carbs like glycans and plant oligosaccharides and fucose and mucin, so that Bacteroides can eat it. Interestingly, Elaine Su from MIT, she took the exopolysaccharide, which is known as PSA in Bacteroides, polysaccharide A. She created an autistic mouse model, and she improved the gut of these autistic mice, rodents, and they even saw autism reversal. So this is mutually beneficial. A lot of symbiont happens. So Julian Tapp in 2009, his group showed a phylogenetic core exists. He had 17 Spanish and Netherlands gut microbiotas he analyzed, and there was a core seen. And with Bifidobacterium, it appeared in 75% of the individuals. Faecalibacterium prausnitzii was in 16 out of 17 of them. So his study, along with others, especially in centenarians and supercentenarians, which are 105 to 109, they have a core microbiome. I call these the ABCs, but it starts with A, Akkermansia, B, Bifidolongum, always, and then a couple Cs, like the Clostridiolas that make butyrate and Bacteroides vulgaris. So over time, the babies have 70% Bifido in their gut. After initial colonization, it flips, and then it becomes only about 7% or less. And then into older age, it goes much down lower. So Ku and his group, he wanted to postulate, well, how can we fix this? You know, babies have such a high amount of bifidos if they're healthy babies. They have such a high amount in their stools, but when we go into adulthood, they as we age, they lose 10 times, and then they lose by older age, elderly, 100-fold difference. How can we bridge this gap for health? Because we know it's so important. So he classified probiotics, and if we are going to biohack bifido, we might want to consider multi-strain and probiotics from different categories that have different roles. So Bifidobacteria, when I formulated it, it's 7-strain. It's histamine-free, so it's tolerated really well by people with allergies, and lactate-free. And it's a high proportion, 5 species of oxalate-degrading bacteria. Oxalates are really quite a problem. A lot of studies like IBD and others, mold, asthma, there's a high amount of oxalates in the system, CKD, kidney disease, anything where there's calcification, heart disease, hypertension, diabetes, fatty liver, fatty pancreas, fatty heart failure. There's a high amount of fungus in these conditions, above normal on average. And these fungi, like oxalates, are produced by candida and mold. So this is a great product that helps to combat it. And there's a lot of extinctions going on. So I'm going to go over one case. I have 3 others, but if you email me for slides, I'd love to send it to you. This gentleman, he did a mini protocol. He had a skin condition for many decades, hidradenitis suppurative, HS. I can't pronounce this too well. This is an old 16S kit from 10 years ago. The company doesn't exist anymore. But initially, he had very low bifidos. Bifidal longum was undetectable, 0. But his actino came up after a protocol a little bit. I actually I did a 16S also 10 years ago, and I did have some bifido, but not much. And that's why I created the probiotic. And he did a mini protocol, some antifungals, some parasite treatment, and then we also did an organic acid test. After treatment, his organic acid test, there were a lot of green zones, and things got a lot better there post. And his protocol used a lot of herbals and probiotics that were bifido and lacto-based. And the second case, we did 16S stool kit testing, 16S, and initially there was no Christian Sinella or Akkermansia detected, and bifidobacterium longum was very, very low. If you look there, it was only 0.0025, but it went up 15-fold after treatment. Again, we used probiotics, and he ate a lot of fermented foods. He added a whole bunch of bionic fiber. I call it bionic fiber, but it's a mix of different prebiotics. And he improved his diet with kale and rice and cook-resistant starch, like a lot of cook-resistant starches. And what he stated was that he had better performance. He was a Spartan racer and an athlete. He felt a lot better. He had less bloating and many other benefits. In the other case, he had lost, I think, 20 pounds. Actually, it's so tiny, I can't see it, but about 15, 20 pounds, and his skin cleared up. I have a couple other cases, but if you email me, I'll send you the slides.